and Son, and the Holy Ghost. Amen. Saint Michael the Archangel, defend us in battle. Be our protection against the wickedness and snares of the devil. May God rebuke him, we humbly pray. And do thou, O Prince of the heavenly host, by the power of God, cast into hell Satan and all the evil spirits who prowl throughout the world, seeking the ruin of souls. Amen. Our Lady, Queen of the Angels, pray for us. In the name of the Father, and the Son, and the Holy Ghost. Okay, so now we get to one of my favorite parts of the conference, which is biology, because I am a biologist. And I think most of you heard my introduction yesterday, or Monday, so I won't super repeat myself. But just a quick recap. I am currently a science teacher at St. John Paul II Catholic High School, where I've been for the last 4 years. This is the conclusion of my 6th year teaching high school science, mostly biology, a little bit of chemistry, a little bit of microbiology, and a little bit of anything else that needs to be taught that particular day, as happens in all Catholic schools. And I have a little bit of a background as a researcher. I'd spent some time at the Genome Sequencing Center at WashU, some time on vaccine development, and some time looking at genetics of transcription factors in C. elegans. So it's a little bit about who I am and what I do. So the talk I've been asked to give today, or the first talk, is titled The Icons of Evolution. And I thought it was important to start out with an understanding of what I mean by icon, because we usually just jump right into the science, and I think the language here is really important. And I'm borrowing heavily, and by heavily I mean like entirely, from a homily by Father Wolf on the icons of evolution. And so he talks about, well, what is an icon? And of course, this is an icon of the creation of Adam on the 6th day. All right, and he describes an icon as not only a sacred image, but a sermon without words. And as we look at these icons, they teach us something, and they teach us something very, very important. And as we continue to gaze on these icons, that truth sinks deeper and deeper and deeper into our souls. So keep that in mind as I talk about these icons of evolution, because they're also images, not sacred. But as we view them over and over and over again, especially without any kind of critical understanding of what they mean or how they came about, those truths do sink deeper and deeper into our souls. So I'm going to use this as a specific example. This is an image that is taken from the American Museum of Natural History in New York. This is right outside their hall on human evolution, and engraved in stone over it says something about human evolution, which I'm forgetting because I didn't write that part down. But Father Wolf points out some very important things about this icon as opposed to the icon we just saw of the creation of man. Of course, it's dark. There's no light. No God the Father. Instead, there's a female gorilla. No tree of life. Just a dead branch. And it's a woman's skeleton receiving the gift of life from a female gorilla, rather than Adam receiving the gift of life from God the Father. No grace, just nature. No life, just death. It's kind of hard to believe anything other than that this is a deliberate and blasphemous mockery of Genesis. And this is the kind of thing we're up against. All right? So I'm going to show you about 7 or 8, I don't remember exactly, I think I split one of them in half. Icons of evolution today. John's getting very nervous because I've highlighted human evolution on the slide. He says, well, that's my talk, and it is, but I'm talking a little bit about genetics and chromosomal fusion. But I'm going to look at some of the things that are presented and by our good friend Ken Miller as some of the foremost proofs, if you will, of evolution. And we're going to look at the science behind those, and we're going to look at why maybe the icon is not as accurate as it should be. So first icon, Hugh twitches when I call this the tree of life, but that's what the evolutionists call it. More accurately describes the tree of death. It started way back there, kind of in the middle. This is where you see the vague printing that says earth, birth, with a single cell. That single cell divided, it became a different kind of cell, and those cells gave rise to other kinds of cells, and amoebas and fish and birds and all sorts of things, is what this icon is supposed to represent. And it seems to represent it pretty clearly. So at the end of every branch there, you have a different species of animal, and this is supposed to account for how all these animals came into being. And in case you're wondering, we're not actually on there, but Neanderthals are. So, I don't know. So, this type of icon is called a phylogenetic tree, hence the tree of life, right? And what you're seeing on the screen right now is an image that's actually frequently used in high school biology classrooms to teach students about how to arrange a phylogenetic tree. So what they would be given is the image that is over here closer to me, which is just sort of a scoring graph showing you which animal has which characteristic. So the leopard is the only thing that has hair, and the lancelet is the only thing that doesn't have any of the characteristics that are being looked at in this tree. So the students will be given these characteristics, they'll be given these organisms, and they'll be said, okay, organize them according to their evolutionary relationships, all right? So clearly, to the evolutionist, the leopard belongs on the outside because it's the only one with hair, and the lancelet belongs on the outside on the other side because it's the only one that doesn't have any of these characteristics. And then as we lose more and more and more characteristics, we get sort of further and further and further up the tree. Is that making sense? Because this can be kind of complicated.
No.
No? Try again? Yes? Sure. Okay, so what evolutionists try to show in a phylogenetic tree is how traits were gained or lost, right? Because they have to account for all the different traits that organisms have through evolution, or it doesn't explain the diversity of life, right? So, I'm sorry? Okay. So everything that has vertebral column, now I do need a pointer, I should have just accepted it, Nick, I'm sorry. Everything that has a vertebral column, see where the vertebral column is over here on the far side? The vertebral column is supposed to have evolved at that point on the tree. So everything after that has vertebrae. All right, jaws evolved next, so everything after that has jaws. So what this tree is showing us is, there we go, these guys are all more related to each other than they are, say, to this guy. Okay, and that's based on these different traits. So because they all share jaws, they're all more related to each other than they are to the guy who doesn't have the jaws and doesn't have the vertebrae. Does that make more sense now? Okay, so my students are asked to construct one of these using just this data, which, as you just discovered, is kind of confusing. And frequently what they'll ask me is like, well, I don't really know exactly where these things go. And especially, often now, even on the AP biology test, they're given this tree structure. They don't have to draw that because they discovered, much to their chagrin, when they rewrote the AP biology exam, that students simply could not draw the tree of the phylogenetic tree, because they had no idea how long ago that ancestor was supposed to have lived, or exactly how those relationships were supposed to have been, based on this limited amount of data. So these students have struggled with this for years and years and years and years. And that's only part of the story. The rest of the story is the fact that you'll be presented with an image that looks something like this in a textbook. So this is a larger tree than what I just showed you. And you'll be shown all these interrelationships and where all these things are supposed to have evolved. But they won't tell you where the real data is, okay? Because you look at this and you think, okay, there's an organism here, and an organism here, and an organism here, and an organism here, right? The only organisms on this tree are up here. That's it. The known identified organisms go at the end of the branches. Everything else is speculation. So when you look at that icon, you're looking at somebody who can arrange things according to characteristics, but has no actual evidence that they're interrelated. And just to address one small misconception from last year, clearly giraffes did not evolve from horses. They came about this way and are related to giant moose, because that makes a lot of sense, right? Okay, this is very related to my second icon, which is whale evolution, because an evolutionist will claim, well, we've got transitional forms. We've got some things at those nodes right there, okay? And this sequence is presented most often as a sequence that has no missing links. It's complete. And the trial that John referenced in his opening talk, where I believe Ken Miller gave evidence to that trial, is that correct? Yeah, and he unveiled at some point in a very dramatic fashion an image that looked a lot like this and said, well, you guys claim we have no transitional sequences. Look, here we have these beautiful transitional sequences. And he showed this icon, okay? So apparently whales came from this little tiny tiger on speed that evolved into a sort of giant swimming mouse that then evolved into something that had sort of smaller legs and a bigger tail, and on down until we have a humpback whale. But there's a couple of problems with our missing links here. And I didn't put any text on this slide because I feel like this picture is a little bit self-explanatory, but these are both artist renderings of the same animal. And I don't know about you, but I think they look awfully different. Like they might not even be related at all. And I'm putting this up just to show that while the people who work in paleontology do have a lot more knowledge, certainly, than I do about how bone structure would indicate, sort of muscle attachment and what the organism might have looked like in real life, the reality is that they can't actually know everything. And you can sort of fudge a little bit. You can build kind of what you want to build just on a skeleton. So when you're looking at the fossil record, you're looking for transitional forms, it's very easy to sort of create the kind of transitional form you want based on the kind of bones that you happen to find. So this is the actual skeleton. This is the most complete image of an actual skeleton of a Pakicetus I could find. But this is what you'll see in the museums. Okay, and this particular reconstruction, not all reconstructions are this disingenuous, because some of them have sort of dark shaded areas that show you where the bones are missing. And I remember one of the first times I ever doubted evolutionary theory was when I was walking through a human evolution exhibit, and I was looking at all the skulls, and most of the skulls had more dark areas on them than they had light areas on them, indicating to me that there was more of the skull missing than actually present. And I didn't want to base my belief in a supposedly scientific theory on something that was basically reconstructed from someone's imagination. So that's our first missing link. This is the formerly mentioned giant swimming mouse. I didn't have pictures of the tiger on speed, I'm sorry. Okay, our first missing link has another problem with it besides possible incomplete reconstructions or conflicting reconstructions, and that's the fact that it lived at the same time as its derivative species. So this is not supposed to happen with two different species that are occupying a similar ecological niche. That's very techno-speak, but that basically means they live in similar areas and they consume similar kinds of preys, and they perform similar functions within the entire ecosystem. So if they're living in the same niche, the idea of the survival of the fittest and competition and all that is that only one of them is going to be able to successfully occupy that niche, and it will gradually outcompete the other one, and the other one will die out. Now, granted, these guys both lived 50 million years ago, but there's a lot of change between these organisms that had to happen in a very short period of time, and they had to coexist for an extended period of time, which is a problem for this evolutionary icon. But this is not something you're going to be presented when you're just shown the icon that I showed you on the first slide. This is the actual skeleton and the reconstructed skeleton of the second missing link, the Ambulocetus. Not too bad here. We have a mostly complete skeleton. This is a pretty good reconstruction. But again, we have the contemporary status issues with Pakicetus. Our third and fourth missing links present another kind of problem. So this is supposed to be one evolutionary step from Rodhocetus to Basilosaurus. Now, I've scaled them for you appropriately, and it was very convenient that my little scale bar down here had exactly 10 feet because I could measure him against my little scale bar, so he's exactly 10 feet long in relationship to that diagram of Basilosaurus. So I don't want to know how that guy gave birth to that guy. That is a big baby. Okay, and in addition to it being a tremendous size difference, which can't be explained by a very small number of mutations, there are other problems. The nostrils have moved significantly. This guy has lost his back legs and his tail is extended significantly, and his entire body is different because he won't be semi-aquatic. He's going to be entirely aquatic. Okay, this is too many changes for a single evolutionary step. So we're missing some missing links, which is probably why we call them missing. Okay, this is a close-up of Rodhocetus. This is one of the more famous finds because it was supposed to be very transitional. And this was I think it was maybe the original artist rendering. Notice the webbed feet. You know, he can swim in the water very well, but of course he's not entirely aquatic like a whale. Notice the actual skeleton. We have the top half of the skull, half of the pelvis, a couple of vertebrae, and half of the leg. You see the webbed feet? I don't either. There's an awful lot of artistry in that reconstruction of Rodhocetus. And it's the artistry that's allowing it to be inserted there in the icon and be a transitional form between the tiger on speed and whales. Okay, so I like to put jokes in my presentation. So sometimes the problem is just that a lot of the pieces are missing in the skeleton. So this is a new icon that's been presented. The other one was a little bit more juvenile, I guess. This one is showing the tree of life along with all of the different animals that are supposed to be transitional forms. And you may notice the Basilosaurus, the giant 66 feet long guy, is no longer there. They substituted Durodon, but Durodon is very similar in physiology to the Basilosaurus. And this icon, even though it's been adjusted and is showing you supposedly a little more information than the last one, still suffers from the problems of the fact that there's no scale, and we have two animals here that are occurring at roughly the same time, and they're not even charted as occurring at roughly the same time. There's now Pakicetus in between Ambulocetus and Rodhocetus, and yet Ambulocetus and Rodhocetus lived at the same time. So there are all kinds of problems with this icon. And that brings me to, well, what is this actually telling us? Well, it's telling us that the scientists can take things and arrange them based on size, color, tooth number, or some other physical characteristic that they can quantify, but that doesn't actually prove anything about evolutionary relationships between these organisms, because obviously, I mean, otherwise oh, I just forgot what he's called. It's not a St. Bernard. It's something else. It's a Bernese mountain dog.
Mastiff.
Bernese mountain dog would not have evolved, or would have evolved from a Chihuahua. So this kind of icon doesn't prove anything about evolution. It only proves you can sort things and that you have metaphysical assumptions. The third icon I'm going to talk about today is something that Father Hickey referenced, and I'm sorry, Father, you did say it backwards. It's embryology recapitulating phylogeny. So the idea here is that if you look at organisms while they're developing, so you go way back into embryonic development, there are some distinct similarities among organisms, and these distinct similarities in our development indicate that we came from a common ancestor, because otherwise we wouldn't have similar developmental pathways. And this particular image is, of course, of a chicken and a human, and I'm very glad that they labeled it correctly. I took this from a Pearson Education publication in 2008. And prior to the early 2000s, what's labeled up here as the pharyngeal pouches were labeled gill slits. When you were developing in utero, you never had gill slits, nor did you ever breathe in water. You got your oxygen through the placenta from your mother. So these at least have been are correctly labeled, but at the time, the idea of gill slits sort of reinforced this idea that when we were in utero, we went through a fish stage, and then a reptile stage, and then then finally a mammal stage, and then finally became human. And this is one of the early justifications for abortion, because it wasn't human until a certain period of development. Because during development we would recapitulate or repeat this whole evolutionary sequence. So we would be less and less and less, or we start out a very unevolved state, and then we become more and more and more evolved until we finally became human. And this is the research that that icon was originally based on. And this image will still appear in a lot of science textbooks. This is Haeckel's original drawings of some embryos that he examined from a fish up through amphibians and reptiles, all the way to mammals and humans. And that's supposed to be a baby human down there. I don't know about you, but that doesn't really look particularly human to me, and nor does the thing next to it look particularly like a rabbit. But so that may lead you to doubt his artistic abilities somewhat. And you would be justified in doing so, because while Haeckel claimed that the top is sort of the earliest stage of embryonic development, and that everything looked very similar there, scientists have since actually taken photographs of embryonic development at the stage that Haeckel identified, and you might notice that the actual photographs look significantly different from his artist renderings. So it turns out that this original icon was actually forged. He took one embryo and sort of repeated the same drawing over and over and over with slight variations, and didn't actually represent what was going on at the stage of embryonic development. And as Jonathan Wells points out in his book, Icons of Evolution, he didn't actually look at the earliest stage of development either. If you go all the way back to the earliest stage of development, you are a single-celled zygote. You make one cell division. Now, how complicated is one cell division, people? It's not terribly hard in the grand scheme of building an organism, but yet you divide differently than a sea urchin divides, which divides differently than a lot of other organisms divide. So even that very first cell division, there are tremendous differences that happen inside the cell, in the way the cell divides, the plane the cell divides, whether it divides evenly or asymmetrically. So you go all the way back to the beginning, the very first division, and we're different from that very first moment. And this is actually the stage where we look the most similar, and we don't even look that similar here. Okay, so embryology does not show common ancestry. It shows a tremendous difference between organisms. Our next icon is homology. And I've defined here homology as structural or behavioral similarity that is observed between two different organisms and arises from a shared common ancestor. And I pulled this definition from a biology textbook, but I'm not at the moment remembering which one, but it's a fairly standard definition used by the evolutionists. And they'll show an icon that looks something like this, where you have a human hand, a cat paw, a whale flipper, and a bat wing. And the bones are conveniently color-coded to show you that your fingers are very similar to the bones in a whale's fin, which are very similar to the bones in a bat's wing, and very similar to the bones in a cat's paw. Right? And so all of the purple bones are thought to have a common origin, all the blue bones are thought to have common origin, that kind of thing. Right? So these structures are said to be homologous because we all have similar sort of digit structures because we came from a common ancestor. There's a problem with homology that's not, it's usually addressed as kind of a side note in the textbooks mentioned, but not gone into very great depth. And that's the problem that we also have structural behavioral similarities between unrelated species that do not share a common ancestor, because these forms share a common function. And one of the examples that's commonly given of this is a bat wing, a butterfly wing, and a bird wing. No one in the evolutionary community is going to argue that a bat, a butterfly, and a bird shared a recent common ancestor, which gave them all wings, especially since the butterfly's wing doesn't even have any bones in it. And if the bird's wings, if you could see the bones, they would kind of all be along the top. There would be no sort of branching finger bones down like the bat wing. So sometimes similarity is homology, but sometimes it's analogy. So this has led to the concept of convergent evolution. And I pulled this image from the Science Times and part of the New York Times from 1998, where they point out that nature discovers the same design over and over again, and that doesn't seem to raise many eyebrows for them, that these things would evolve not once or twice, but 3 or 4 or 5 or 6, or in the case of the bacterial flagellum, at least 5, if not 7 times, independently of each other. Now, why is this a problem? Well, even evolutionists admit that the odds of developing complex structures are fairly low. The odds of developing them twice are much lower. The odds of developing them 5 times are infinitesimally low. And something like the bacterial flagellum, which contains 40 different proteins, And each of these proteins is present in multiple copies so that you have a structure that's thousands of proteins all together, and these all have to be coordinated together to do a very complicated task, which is to basically turn like a little motor and propel the bacteria through whatever medium it's swimming in. For this to have evolved 5 to 7 times, and this was reported in 2009 in Trends in Microbiology, doesn't make a whole lot of sense. It would make a whole lot more sense that at one time the flagellum evolved, and then everything that had a flagellum sort of originated from that species. But when they do other studies, some genetic studies, it shows that actually, based on the mutations, these species are not as closely related as we think they are. So we had to have all these different incidences of evolution of the bacterial flagellum. And I know Michael Behe quotes or cites this often as a an example of irreducible complexity. So something that is so complex it could not have evolved stepwise. And wasn't really planning to deal with that argument today, but maybe I'll put Rick on the spot and ask him to say something on it. But something that's this complex, it's not a simple structure. It's something that it's very hard even for evolutionists to imagine how it came about stepwise entirely. They've got a few ideas for what might be possible transitional steps, but some of those look more like a flagellum that degraded than a flagellum, or something that was built up into a flagellum, which I will address in my next talk. It defies reason. And as Father Tom said, when you start to believe these things, you have to believe things that are very unreasonable. So genetics has kind of replaced, or is beginning to replace, the study of physiological structures in homology, because you have a whole lot more base pairs in your DNA than you have physical structures to look at. So it gives the evolutionists more data, so they can look at all of the base pairs in a DNA sequence, and what you're seeing here is sequences that are highlighted that are similar across all 12 of the different species, or identical. The blue ones are identical across all 12 of the different species. These base pairs would be called conserved, because they're present in a number of different organisms, and it's thought that, well, the original ancestor had a sequence that would have had all of these things that are highlighted in it, and then would have had maybe some different bases in the part that is not highlighted, and this is actually not a DNA sequence, but a protein sequence. I'm just trying not to be super confusing to the non-biologists in the audience. But the closer two things are related evolutionarily, so the story goes, the fewer the differences there will be between them. So the ones that have the most base pairs or the most amino acids in common in a DNA or a protein sequence are thought to be more closely related. And then the ones that are a little bit more different, they're going to be further out on the phylogenetic tree. So we'll draw them a little branch over here. And the ones that even have more mutations, we'll draw them another little branch over here. So the branch will be further and further away. So this seems like a good idea, except that it leads to some really interesting conclusions. One of which is that you share half your genes with a banana. Okay. You also have the same hemoglobin that's found in plant. Now, hemoglobin is the thing that carries oxygen throughout your body. So clearly you and plants shared a common ancestor recently, and never mind. Rabbits, as of a 1996 analysis of about 88 proteins, are apparently not rodents, they're primates. So, they're more closely related to monkeys than they are to anything else with long teeth and, sea urchins are chordates. A chordate is something that has a backbone. Cows are more closely related to whales than they are to other hoofed animals, like horses. Sharks and camels are closely related. And bats and dolphins, too, because their sonar system is identical on a molecular level, or at least almost identical. So when you start looking at genetics and looking for similarities, you come up with some really wonky stuff that doesn't agree with the rest of your wonky stuff. Wait, did I just call them both wonky? And of course that leads to the conclusion that bunnies must have come from tomato plants. And I've got photographic evidence. Okay. So I would like to put forward an alternative explanation for these similar structures. And I'd like you to raise your hand if you think you know what is on the screen.
chair.
Yeah. Okay. Some of you don't raise your hands very well. It's been a long time since you've been in school. Of course, I have this problem with my teenagers as well. But yeah, they're both chairs. How do you know that? Well, they have a structure that's similar to a chair, even though neither one of them is like any chair I've ever sat in. There's clearly a back, there's clearly a seat, it's clearly made to support a human form while sitting. Structure is related to function, not to common ancestry. And any biologist worth their salt will emphasize to you again and again and again and again that structure is intimately related to function. Yeah. The way that something is structured is going to determine the way it functions, especially at a molecular level. Okay? We see this particularly with enzymes. Enzymes have to be folded properly in order to work. In fact, if you've heard of mad cow disease is caused by an improperly folded protein. So the protein doesn't form the correct three-dimensional shape, it forms an alternative three-dimensional shape. And normally, a defective protein like that would just go sit in the corner of a cell and be defective until it was degraded by a protease. But in the case of prions, they actually bump into other proteins that are correctly folded and cause them to unfold and become prions. So they're like, Teenage Mutant Ninja proteins or something, I don't know. Okay, but the way that it's folded determines whether or not it's functional. And enzymes have a particular spot called an active site, which you can see up here in the diagram. That active site is necessary to bind whatever the enzyme's going to act on. If the enzyme can't bind its substrate that it's acting on, it can't do its job. It can't do anything. So if you had an enzyme for digesting, say, lactose, and it couldn't bind the lactose, guess what you can't do? You can't digest lactose. That's actually not why you're lactose intolerant. I'll get to that in the next presentation. Okay. But any change in this active site is going to affect the binding of that substrate. It's going to affect the function of the enzyme. Okay. So why are all these sequences so similar across all these animals? Well, if you're an animal, you have to breathe. So you have to deal with oxygen. Oxygen can be kind of toxic and can damage your DNA. So you might need some proteins that can deal with DNA damage and repair. You might need some proteins that are involved in the electron transport chain that's going to work very similarly across all organisms. You might need some proteins that are going to allow you to digest the same kinds of sugars everything else eats. These proteins are similar. These DNA sequences are similar because the proteins are similar. The proteins are similar because they have to have the same function. Okay? And a very, very small change in shape at a molecular level can make a big change in the functionality of the molecule. And we can see this here in starch and cellulose. So if you consumed any of the tortillas at lunch, you ate some starch today. And you can digest that. You can use that as an energy source. If you consumed some grass, I don't know why you would do that, but you would have eaten cellulose today. You can't digest that. Okay, and you can't digest that because of a tiny change. This is a glucose molecule in starch. This is the same molecule in cellulose. Glucose and starch and cellulose are both made entirely of glucose, but they're bonded together in different ways, right? This has one kind of linkage, this is another kind of linkage. And usually I ask people to comment on this, but I forgot this time. So I already pointed out, so I can't give you a pop quiz. But basically, these glucose molecules are upside down from the ones that come before them. So in cellulose, they're sort of right side up, upside down, right side up, upside down, and starch, they're all right side up. And whichever side is right in molecular space, I'm not really sure. Because of this tiny change in shape right here in this bond, you can digest starch, and you can't digest cellulose. It's the same chemical formula. It's made of the same subunit, glucose. And you can digest the one, you can't digest the other one just because I flipped it upside down. Okay, tiny change in shape, big change in function. So a lot of this conservation that we're seeing across species is because if those things weren't the same across species, those proteins would lose their functions. Next icon, vestigial structures. When vestigial structures were first proposed, it was thought that the human body contained 180 useless relics of evolution. These are structures that sort of performed no function in the body now, but they must have had a function at some time in a previous common ancestor, and so we were just sort of loaded with these evolutionary relics that had no function. it turns out all 180 of these evolutionary relics have now been identified as having some kind of function. Okay? Raise your hand if you can wiggle your ears. Ah, yes, there's always someone. Always Rick. And Daniel. Okay. That doesn't seem to be a particularly, shall we say, necessary for survival skill in modern America, or probably even in modern Uganda. But if you can move your ears, you can sort of change the way that you're picking up sound, which can allow you to pick up sound better in the environment. Now, we can't so much do that anymore. Most of us have lost the ability to move our ears, but that's not because those muscles didn't have a function and they didn't contribute to hearing better. It's because if you don't use it, you lose it. This is true of language and also of muscles. If we're not continually exercising our muscles, we're not going to continue to be able to move them. All right? And I'll get in my next talk to a concept about the human body has been degrading over time, not building up over time. And so it's, even if there were vestigial structures, it wouldn't actually really be that great of an argument for evolution. It would be a much better argument for the fall. That at one time we were completely fully functional, and now we're gradually losing function because we're gradually getting further and further and further and further from perfection. Wisdom teeth, which most of us have had extracted, in people that eat a sort of more indigenous healthy diet, their mouths actually accommodate their wisdom teeth. They don't need the extraction, and they're good to go. They're functional teeth. The human pelvis, the tailbone, is an important site for muscle attachment. Some of the muscles that are involved in walking upright actually are attached to your tailbone. And the appendix, last one on this slide, is a necessary part of your immune system, especially from 0 to 2 years old. And if it wasn't present during that time, you would have a much harder time developing a healthy immune system. Now, just because you can remove it later doesn't mean it's not useful. You can cut my arm off, I will survive. But I'd really like to have both arms. And I'd also like to keep my appendix. Okay, ostrich wings are my favorite vestigial organ. Of course, everybody says, well, ostriches can't fly, therefore they don't need wings, therefore, here's, even if we can say these human organs have a function, there's other organs that don't. Well, it turns out that ostriches have an amazingly intricate network of blood vessels on the underside of their wings. Okay, so when an ostrich flutters his wings, all of that blood that's in those blood vessels is exposed to the outside air. And where ostriches live, it's very hot during the day, it's very cold at night. So the ostrich is warmer than the air usually, and so when the ostrich flutters its wings, that blood is exposed to the cooler air. It actually cools down. The cooled blood from the wings goes back into the ostrich's core, keeps the ostrich from overheating and dying. Now I don't want that at nighttime because it's cold. I don't want to be cold, so the ostrich just tucks his wings up, turns off his air conditioning. It's an incredibly well-designed system, not a useless structure. Okay, next icon. We're 98% chimp. I'm sure you've seen this somewhere before. So the claim is that our DNA differs from humans and chimps by only about 2%, so clearly we're closely evolutionary related. But let's back up here and look at some actual data. Chimps can't play chess. More accurate estimates are going to give that dissimilarity at between 3.9% and 13%. And this is stuff that's been published in Nature in the Proceedings of the National Academy of Sciences. So these are not creationist articles I'm citing. In fact, I'm only citing things from evolutionary biologists for the most part. And if I'm not, I promise I'll tell you. So if you multiply that by the number of base pairs that we have as human beings, that's 117 million base pairs. That doesn't sound quite as insignificant as 2%. So if we put it in perspective, if we printed all of this DNA onto a 8.5 by 11 piece of paper, it'd take about 29,000 pages to record all the differences between you and a chimp. 29,000 pages. That's more than Dr. Syngenesis' book. Sorry, I only teach you about like you. Okay. I know, right? So given that evolutionary estimates place a divergence or a separation between chimps and ourselves about 300,000 generations ago, that number of differences would require an average of 390 mutations per generation. Now, that's not 390 mutations in your entire body, that's 390 mutations in your germ cells. So specifically in the egg and the sperm cell. That's a lot of mutations for one cell to accumulate in one generation. And at that mutational load, we really should be mostly dead. But mostly dead is slightly alive. Okay, if you look at other things besides just total genome sequencing, human-chimp differences are even greater. So miRNA stands for microRNA, and if you've heard of RNA, it's the copy that you make of the DNA, in the messenger RNA is anyway, but I won't get too complicated. But it's a copy that's made of the DNA. Sometimes it has some enzymatic functionality, sometimes it's used as a copy to make proteins, carries instructions, and microRNAs are actually used to regulate other pieces of RNA. So they will bind to the other pieces of RNA, cause them to be degraded at certain times, so that you're not making proteins you don't need when you don't need them. So they just looked at microRNA expression in the brain, and there are only 447 microRNAs that have been discovered. Chimps had 25 that we didn't, and we had 51 that they didn't. So if you do the math, we're 17% different just in our microRNAs. And the microRNA part is part of the regulatory system of the DNA. So to go back to my comment earlier about structures being similar because they had a similar function, our genes are similar because they have to do similar things. Our regulation is incredibly different. Our regulation is incredibly different because our DNA has to build a human, their DNA has to build a chimp. We both still have to breathe. So we both need proteins that deal with oxygen. But we have very, very different regulatory sequences. Okay, another major genetic difference, telomeres are something that occur at the end of chromosome. So when DNA replicates, so when it makes a copy of itself, so every time your cells divide, the chromosomes have to be copied. And because of the way the DNA polymerase works, it can't copy all the way to the end of the chromosome. It's going to lose a little bit of information at the end of the chromosome. So it's thought that these telomeres, which are just sort of regions of repeated DNA, so sort of like the record skipping, if you will, so just, the same sequence over and over and over again. If I lose a little bit of that, it's not going to damage my ability to continue to continue to build cells, because it's not information that's a gene, it's not information that's regulatory in nature, it's just sort of like a cap to kind of protect the end of the chromosome. Our telomeres are very, very different from chimps. They're less than half as long, which is interesting because people also associate telomere length with lifespan of the cell. So it's not that once a telomere runs out, then the cell can't divide anymore. Yet we live a whole lot longer on average than any species of great ape. Some other major chromosomal differences are chromosome 4, 9, 2, and Y are incredibly different from chimps. And we have 689 genes that they don't, and they have 86 genes that we don't. So again, if we're looking at sort of the more important aspects of what exactly this means, not just the sequence of base pairs, but what genes are present, what regulation is present, how are our chromosomes organized. There's differences here that evolutionary biology can't explain if we're closely related only 300,000 generations ago. But that's assuming all that, even all those problems, are assuming that the chimp sequence is correct. But I just recently learned that the chimp sequence has some interesting problems with it in itself. So this diagram probably means nothing to you. I'm sorry it's not labeled, but it was the best I could find to explain shotgun genome sequencing. So I worked in the Genome Sequencing Center at WashU. And what you do when you're sequencing a genome is you take the whole genetic information of the animal and you chop it into little random bits. And it helps if they're roughly the same size. So that was my part of the project. I would take the DNA and cut it into roughly 2,000 base pair lengths. Okay, and then you would grow this, you would transform bacteria with this DNA. You would grow that bacteria, you would extract the DNA out of the bacteria, you would sequence the DNA out of the bacteria, and by the way, at every point in this process, there's a possibility of an introduction of an error through mutation, because the rates of fidelity of enzymes in the lab are less than the rates of fidelity of enzymes in your body. So it's possible that there are errors in the sequence, but given that we're collecting lots and lots of reads of the same area, we're hopefully eliminating that because the errors aren't likely to occur at the same space every time. So that's kind of what you're seeing at the top is the whole genome. It's complete. The next line is cut into pieces. And of course, these pieces are not in sequence when you first get the sequence of them in the laboratory. So then a whole other team of computer scientists has to go through and say, well, where do the pieces match up? So where are there some overlapping ends? And wherever there's an overlapping end, I can say, okay, this piece came after this piece, or before this piece, or next to this piece. And the more overlap I have, and the more pieces I have, the more I'm sure that I'm correcting or constructing the correct sequence. And this is how the human genome was sequenced, and it took about 15 years. The chimp genome, they decided, well, the shotgun method's a little, labor intensive and it takes a long time. It would really help if we had a scaffold that we could sort of, compare and assemble this chimp genome against. So guess what they did? They used the fully constructed human genome sequence as a scaffold to develop the chimp sequence. So all these similarities that we're seeing may not even be real. They may be artifacts of the way this thing was sequenced. And at this point, even evolutionary biologists admit this chimp sequence is not very good. They're resequencing it correctly this time, not using a scaffold. But the fact that they use the human genome as a scaffold by itself invalidates all of this data and all of these conclusions. Because it's similar because I constructed it using the first one as a pattern. Not cool, guys. Not good science. All right. Next icon, also humans and chimps. This is showing chromosomal fusion. So humans have one less pair of chromosomes than chimps do. And evolutionary biologists explain that by saying, well, chimp chromosome 2A and chimp chromosome 2B, they fused together and they became human chromosome 2. All right, remember the telomeres at the end of the chromosomes from a minute ago? So if that happened, then I should have a region of telomere DNA in the middle of chromosome 2, where I wouldn't normally have telomere DNA. Okay? And so evolutionary biologists pointed out, and they said there's telomere DNA in the middle of chromosome 2, and there is. Right? And there's another centromere that's been inactivated, which there's a centromeric-like region, but whether or not it was an actually centromere is questionable. But again, there's some problems even with this interpretation, because there's only around 185 telomere repeats at the region they've identified as where the two chromosomes should have fused, and there should be about 5,000. So what that means is that if there were telomere repeats before, they have mutated at a rate that cannot be accounted for by the rate of mutation proposed by evolutionary models. So this thing has degenerated or mutated much, much, much too fast for this to actually be a fusion site between chimp chromosome 2A and 2B. Another problem is that since the fusion of the chromosomes was head-to-head, so if they're like this, normally they sort of one flipped upside down and attached to the other one. So the telomere repeats should be palindromic on either side of the fusion site, so one should read in one direction, the other should read in the other direction. On both sides of the fusion site, they read in both directions. That should not have happened, it should not be, if they had actually fused. And there are actually probably 10 or 15 problems I could go through with the telomere fusion, but some of them are more technical than others. So I'll leave you just with this third one, which is, I think, the most important as a biologist. These telomere repeats are not in sequence, and they're not in the same reading frame. Now, if you're a biologist, you know exactly what that means. If you're not, I'm going to have to explain a little bit. But when DNA is read by an enzyme, it reads it in sets of 3. Okay, so this particular read that you get depends on which base pair you start on. So you can get 3 reads off of any piece of DNA. And actually, this is one of the things that bacteria do. They will occasionally have one sequence in one frame and another sequence in another frame in the same piece of DNA. It's highly conservative, so they don't have to have as much DNA in their cells, and they can have multiple functionality with one piece of DNA. So the fact that these telomeric repeats, now telomeric repeats are in sequence normally, and they're not in sequence, so the very few repeats they found, is they're not nearly enough to justify a fusion site. They're not reading in the right direction on either side of the fusion site. And if you go straight through in a single reading frame, there aren't even 185 all in the same reading frame. There are 185 in the 3 different reading frames. So on average, you've really only got 60 telomere repeats where you should have 5,000. I don't know about you, but I don't think this is very good evidence for fusion. Okay, I think this is my last icon. Those are words everybody likes to hear, right? In conclusion. It's the Miller-Urey experiment. So once upon a time, a graduate student named Stanley Miller took some gases that were supposed to represent Earth's early atmosphere. So this would have been water vapor, methane, ammonia, hydrogen gas, and some carbon monoxide. He stuck it in a flask, and he stuck an electrode in there, and he sent an electrical spark through the flask to simulate lightning. And he created some amino acids, and everybody got super excited because here was proof that amino acids could form on the early Earth, therefore life could have evolved. I don't know if you're like kind of reading my mind or something, but I might tell you there's some problems with this. So 2 of the problems are huge, and they have to do with oxygen and hydrogen. So based on studies of rock formation, scientists concluded that from as far back as we can measure, the Earth's atmosphere had oxygen. Now, notice Miller's flask did not have oxygen. Do you know why it didn't have oxygen?
Fire.
Well, because if you have oxygen in that flask, oxygen reacts with a lot of things. And it reacts with all those amino acids, and it turns them into goop that's not biologically relevant or useful. Oops. Also, based on other studies, we've determined that there was probably little free hydrogen in the early Earth's atmosphere. If I don't have hydrogen, I don't get amino acids. So based on what was and was not in the early atmosphere, Miller's experiment is not representative of the Earth's early atmosphere as far as we can, figure it out based on geology, which could be argued as maybe not entirely representative, but I don't know enough about that to validate or argue against it. But his amino acids simply wouldn't have formed. And there's some additional problems in the way that he sort of trapped the amino acids. Had they been in an actual natural environment, it's highly unlikely that any of them would have been close enough together to react. And it's also highly unlikely that any of them would have lasted very long because they wouldn't have been in the right environment to persist. But it basically worked. His experiment worked because he took the elements that you need to make amino acids, he put them in a flask, and he made amino acids because he had all the proper elements, except for one. So he had hydrogen, carbon, oxygen, nitrogen. He was lacking sulfur. And now, why does this matter? Only 2 amino acids contain sulfur. Surely that, is not terribly important. Well, it turns out that the 2 amino acids that contain sulfur are 2 of the most important amino acids in protein formation. Methionine, which happens to occur at the beginning of every single protein in every single organism. It's a start codon, people. You can't start a protein without methionine. So how did we get to a life system that starts with sulfur-containing amino acids if we couldn't create sulfur-containing amino acids in this early atmosphere? And also, cysteine is sulfur-containing, and cysteine forms disulfide bridges in protein structures. So protein structures are stabilized by this. So sort of like you're building with Lego blocks. When you're really little, you just build the blocks straight, one on top of the other, and your structure's not very stable. And then you get a little older, and you're like, hey, if I overlap my blocks, then my structure will be more stable. Right? This is sort of like the overlapping of the Lego blocks in the protein structure. So if I don't have that structure, I don't have stable protein structures. So it's very hard to imagine how a stable protein structure could have spontaneously arisen without sulfur, which, to my knowledge, has not been accounted for in the Miller-Urey experiments or the follow-on experiments. And the last problem I'm going to talk about the Miller-Urey experiment is chirality. So molecules have what's called handedness. So one of them is left-handed, one of them is right-handed. And it turns out that in your body, you only use left-handed amino acids. You don't use right-handed. There is no known natural sorting mechanism to distinguish left-handed from right-handed amino acids other than the enzymes that are in your body that are already part of your ability to be alive. So how this could have been separated out so that there were only one kind of amino acids in your body in things that were developing as life was evolving is a huge problem for evolutionary biologists. And in fact, this one icon by itself could completely do away with biological evolution. And any chemist worth their salt will be willing to admit this, because the chemistry is impossible. And evolutionary biologists, they kind of focus on the things that come after that. They focus on the tree of life. They focus on the whale evolution sequence. They focus on the things they can see and the things they understand and the molecules. But you can't get from non-life to life by any chemical method that we can even imagine. So it's just not it's not a tenable hypothesis, but you already knew that. So just to recap, evolutionists' proponents fail because phylogeny doesn't establish evolutionary relationships, it assumes them. The perfect whale sequence is not so perfect. Embryology does not show our similarities, it shows our differences. Homology is a mess. It could be analogy or convergent evolution or anything else that might not be homology. Vestigial structures happen to be useful, maybe designed. We're not chimps. And biomolecules don't form in primordial atmospheres.